Hello, my name is Alec, and today I'm going to share with you my research project that I've been working on this summer through the USOR program. And so its title here is Identifying and Analyzing Abundance of Beetles Collected from the Pennsylvania Hemlock Forest. Here's my abstract, which I will read to you. And so despite the importance of beetles to forest health, the community in hemlock trees has not been characterized. This proposed research seeks to describe the identity and abundance of beetles in at-risk forests as the hemlocks slowly decline from an infestation of the pest insect, the hemlock woolly adelgid. The information on the beetle populations can be a baseline for future work and a backdrop for the development of pest control methods. Beetles have already been collected as a bycatch from 569 traps in central Pennsylvania forests in the spring of 2019, which were intended to monitor the Laricovis nigrinus, which is a biocontrol agent of the hemlock woolly adelgid. While that study is complete, the bycatch beetles aren't about alcohol. The situation invites further work on the beetles and will produce identifications according to abundance, timing, and location, which will advance our knowledge of beetle diversity to enhance monitoring and control going forward. So the depletion of the hemlock forest that's currently going on in Pennsylvania and across a lot of states is due to the hemlock woolly adelgid, which is an invasive pest that came from Asia, and it'll feed on the sap of the trees, which then results in the tree's death. And currently it's unknown if there are any natural predators to the hemlock woolly adelgid in Pennsylvania, but even if there are, they are not able to control it efficiently. So you can see here is just a forest that has been almost completely wiped out from the hemlock woolly adelgid. And here is a hemlock tree that has the woolly adelgid on it, and they produce this white flocculent that protects itself. But then down here in this corner is the hemlock woolly adelgid without its the flocculent. So what is biological control? It is the use of species to control a pest or invasive population. It reduces the need for pesticides, and it's the introduction of a natural predator that allows for the natural ecosystem to continue to thrive. So in the hemlock forest, a beetle called Laricovis nigrinus, or LN, which was collected in Seattle, was released as a biocontrol of the hemlock woolly adelgid. So here in the bottom is the LN larva, and then in the top here is the adult, which is feeding on the hemlock woolly adelgid. And so my personal research, we had 569 vials of insects taken from samples from the Pennsylvania hemlock forest. The beetles were isolated from the samples and identified using a dichotomous key. The number and type of each beetle was collected for each trap in an Excel sheet, and then high quality photos of the photographs of the beetles were taken with a microscope. Photos were then uploaded to bugguide.net for expert species identifications, and for further identification, we had the beetles DNA barcoded, but we have yet to receive the results from the first plate. So then I'm working with two other students on the project, Mary Bland and Alexis Boback, and so far we've gotten through 285 samples that date from April 1st to May 13th of 2019. And over here is just the microscope that I use when I'm working. So then next here is just some pictures from what I guess my work looks like, what I do. So here is just a sample of before the beetles are isolated. So you can see there's a few different larvae and flies in there that we just have to isolate the beetles out of. And this is my full setup when I'm working. I have the microscope, which has a camera which attaches to the computer, so we're able to take pictures with that. And then up here on the double monitor is the Excel sheet, and this is just a close-up of what the beetles look like through the microscope camera. So now I'm going to discuss some of the features that I learned to look for when identifying beetles. First, here we have a firefly, which is a beetle despite its name. We know this because the elytra, which is the hard covering of the wings, here is they meet in a straight line down the middle. And then a key characteristic when identifying the fireflies is that from a dorsal view, which is this on the left, their head is completely hidden by their large pronotum, which is this here. And as you can see, on a ventral view, they have it's a big and it covers their head, but you can see their head there and here from the side, but it's just you can't see it from the dorsal. And so next I have a Carabidae beetle, which are ground beetles. They can be identified by their neuropleural sutures, which are these right here on the middle part, and it's just this lighter part here, it puts them in the family Adelphica, which most beetles are in the family of Polyphaga, but because of the no plural sutures, they get put in a different family. As for Staphylinidae, they tend to have a very unique shape that's easy to identify compared to other beetles. They have these short elytras, as you can see here, it's very short and the wings are exposed, and they just have longer bodies that are easily recognized and just very unique. So here is a bark beetle, which again, is just a very unique beetle. It has a very unique profile that really no other beetles share. So when you see a bark beetle, you're like, definitely it's a bark beetle. There can't be, it would be pretty hard to like misidentify it. So here we have a bunch of different species of bark beetles that we identified. So this one here on the top right is the first bark beetle that we found. So we use it as a baseline for all the other bark beetles. So over here in the left, on the left, is a beetle that we called Lighthead. And as you can see, the head here is just a lighter yellow color compared to the baseline bark beetle. Here in the middle is, sorry, sorry, these pictures aren't great for showing the identification, but they have a small scellum, and it's much smaller than the others, and that is a part here on the thorax at the top of the elytra. So down here in the bottom left is a beetle that we call just the bright bark beetle. You can see it's much brighter compared to the other ones. And then lastly, down here, we have, sorry, we have, so this one is called Small Club, which again, these pictures really aren't great for showing the identification, like the feature. But the club is parts the end of their antenna, and it comes to a big, I guess like it's thin, and then it comes out to a club. But they have a lot smaller club antennae than the other beetles do. So finally, these are beetle larvae, which are pretty easy to identify because they have a developed head, front-facing mandibles, and long legs without prolegs. So they just have these long legs in the front and then no other legs in the back here. So it's compared to like flies and other larvae, they stand out pretty well. So why is this important? Before now, the communities of beetles in Hemlock forests have not been categorized. The data collected can be used as a baseline to help develop pest control methods. Knowing the beetle diversity in the Hemlock forest can help us understand what may be preying on the Hemlock lily delicate. This information may be able to help create new methods of biocontrol. And so over here on the left, you can see a photo, which on the left is a hemlock tree that has suffered from an infestation and is very obviously dying. And then on the right is a hemlock tree that's healthy. So that's the end of my presentation. Thank you for giving me the time. I hope you have a great day. Thank you.